Learning objectives of today's video is to describe and identify the microanatomical features of the kidney. Kidney, it is considered as a part of the urinary system. So the urinary system, we are having two pair of kidney, a pair of ureter, urinary bladder and urethra. So we are having two kidneys. So each kidney, it is bean shaped. It lies in the posterior abdominal wall and it is retroperitoneal, and it measures about 7.5 centimeter in length, 5 centimeter in breadth and about 2.5 centimeter in thickness. Each kidney is having a medial concave border called the hilum of the kidney, through which the renal vessels and the nerves pass. Along with the renal vessels, we are having the renal sinus or the renal pelvis. So this renal pelvis, we also call it as the pelvis of the ureter, and along with its subdivisions. So these subdivisions, they are the major calyces. Major calyces are 2 to 3 in number, and the minor calyces, which are about 8 to 12 in number. The substance of the kidney, we divide it into the outer cortex and inner medulla. So the outer cortex, it seems to be granular in appearance or a reddish brown zone, and the medulla, it is seen as a pale zone, and the kidney, it is covered, or we can say outside the cortex we are having a thin layer of capsule. So if we talk about the medulla, that is the inner part, so it is formed by the renal pyramids. You can see these are the renal pyramids. So these renal pyramids, they are about 8 to 12 in number. So these pyramids, they are triangular in shape, having their base. The bases of these pyramids, they face the cortex, whereas their apexes, they faces towards the renal sinuses. And these apices we also called as renal papillae. These renal papillae, they are cupped in the minor calyces. So at the tip of each of these papillae, we can see a duct, and those ducts are called as papillary ducts or the duct of Berlin, which open into the minor calyces. So if we see the base of each of the renal pyramid, we can see the medullary tissue extending into the cortex, and this part of the renal medulla extending into the cortex, it is called as medullary ray. So similarly, the cortical tissue that extends in between the two pyramids or in between the adjacent pyramids, we call it as the columns of Bertini. Renal cortex, it is granular in appearance and it is formed by the renal columns and the cortical arches. So the cortical tissue that is extending between the two adjacent pyramids, we call it as a renal column of Bertin, whereas the tissue lying between the base of the pyramid and the surface of the kidney, it forms a cortical arches or cortical lobule, and this part of the cortex, it shows light and dark striations. So the pyramid and the cortex around it constitute one lobe of the kidney. Here you can see the area within the dotted line, that is one pyramid along with the cortex that surrounds it. Now from the base of the pyramid, the medullary tissue is extending into the cortex, and we call it as medullary rays. These can be seen as light striation. So the medullary ray and the surrounding cortical tissue, it will form the renal lobule. Before going into the detail of the microanatomy of the kidney, we should know its vascular structure or the vascular supply of the kidney. So each kidney, it is supplied by the branches of the renal artery. So from the aorta, we are having renal artery, a pair of renal artery, one for each of the kidney. So this renal artery, it then gives the segmental artery. So the segmental arteries, now they divide to form the interlobar arteries. These interlobar arteries present in between the two pyramids, they form the arcuate artery. So this arcuate artery, then it leads into the interlobular artery. So the lobular arteries, then they enter or form the afferent arterioles, and then within the glomerulus we are having the efferent arterioles. These efferent arterioles, they drain into the peritubular capillaries, or they may drain into the vasa recta, and then from the interlobular veins into the arcuate veins. Those arcuate veins, they drain into the interlobar veins, and then into the renal vein, and finally into the inferior vena cava. So here we can see this is the arcuate artery. So this arcuate artery, we can see the direction of a flow of the blood in the arcuate artery and then in the efferent arterioles. So the blood direction is almost perpendicular to the blood flow that is seen in the arcuate arteries. So from these efferent arterioles, the blood, it enters into the glomerulus. From there we are having the venous, that is peritubular capillaries are formed, and those together they drain into the arcuate vein. Each kidney, it is made up of 1 to 4 million compactly packed uriniferous renal tubules, and these tubules, they are embedded in a vascular interstitium. So each uriniferous tubule, it consists of a nephron, and this nephron, it is considered as a secreting part, and the collecting duct or the collecting tubule, it forms the collecting part. Each nephron, it is considered as a structural and functional unit of the kidney. The length, it varies from 50 to 55 mm. It consists of renal corpuscle or the Marfilgian corpuscle, which is considered as a unit of filtration, and it consists of glomerulus and the Bowman's capsule. So the Bowman capsule is the outer part, whereas the glomerulus, it is the inner part. The other is the renal tubule, and this renal tubule, it is for the selective reabsorption of the material, and it consists of the proximal convoluted tubule. Then we are having the loop of Henle and the distal convoluted tubule. We are having two types of nephrons. The nephrons that are located in the cortex, they are called as the cortical nephron. They are having short loop of Henle, and they extend only up to the outer zone of the medulla. The other, we are having the juxtamedullary nephron. Juxta means at the complex or near. So they are located in the corticomedullary junction. So they are having the long loop of Henle that extends deep into the inner zone of the medulla, and this part, it is accompanied by the vasa recta. Each renal corpuscle, so it is formed by the glomerulus and the Bowman's capsule. So the Bowman's capsule, so it is considered as the blind ended end of the nephron, which is converted into a cup-shaped structure by the invagination of the tuft of capillaries that we call as glomerulus. So each Bowman's capsule, it is having two layers, that is the outer parietal layer and the inner visceral layer. And these two layers, they are separated by a space, and we call it as Bowman space or the urinary space. The outer parietal layer, it is made up of simple squamous epithelial cells, and the capsular epithelium, it becomes continuous with the cuboidal epithelium or the epithelial lining of the proximal convoluted tubule at the urinary pole of the corpuscle. The outer parietal layer, it is continuous with the inner visceral layer at the vascular pole of the corpuscle. So these cells of the visceral layer, they are modified, or they are specialized epithelial glomerular cells or epithelial cells. We call them as podocytes. We are having two poles, that is the vascular pole through which the afferent and the efferent arterioles enter and leave. The other is the urinary pole. From here the proximal convoluted tubule arises. This urinary pole, it is opposite to that of the vascular pole, and the space between the parietal and the visceral layer, it is called as the Bowman's space or urinary space or the capsular space. Now glomerulus, it is a tuft of anastomosing network of the capillaries that is supplied by the afferent arteriole and drained by the efferent arteriole. The glomerular capillaries, they are lined by fenestrated endothelium. This capillary network in the glomerulus, it is supported by connective tissue cells called as the mesangial cells, and these cells, they are the stellate cells with contractile and phagocytic properties, and they may be involved in clearing the glomerular filtrate. The glomerular capillaries are closely invested with the visceral layer of the renal corpuscles, and those cells, they are called as the podocytes. We see numerous nuclei in the histological section of the glomerulus, and those are of the endothelial cells of the capillary lining and mesangial cells and the podocytes. Podocytes, they are the cells of the visceral layer, and they are having the long cytoplasmic processes, and we call them as the primary processes, which in turn gives rise to short secondary foot processes, and we also call them as the pedicles. The secondary processes, they embrace the capillaries and interdigitate with the secondary processes of the other podocytes, and the cell bodies of the podocytes and their primary processes, they do not touch the glomerular basement membrane. So here we can see these are the podocytes. This is the cell body of the podocyte, this is the primary process, and this is the secondary process. So the secondary processes, they embrace the vasculature of the glomerulus. Here we can see this is one podocyte, and this is the primary process, and these are the secondary processes. Now the space in between these interdigitations, it is uniform, and the width is almost about 25 nanometer, and we call this space as a filtration slit or slit pores. And these slit pores, here you can see, they are covered by fine filaments that constitute the slit diaphragm. Now the filtrate, it does not pass through the podocyte cytoplasm. It is because of these slit diaphragms. The filtration, it occurs through the basal lamella that is thickened at the filtration slits by some filaments to form the glomerular slit or the diaphragm. Now during filtration, what happens? The glomerular filtrate, it passes through the three layers. Here we can see a lining epithelium or the endothelial lining of the glomerular capillaries. Then we are having the glomerular basement membrane, and then we are having the filtration slits between the secondary processes of the podocytes and the slit membrane bridging them. So here we can see the enlarged view. So this is the capillary. So this is the endothelial lining or the capillary endothelium. Then we are having the thick basement membrane. These are the foot processes of the podocytes of the glomerular capsule, and in between the two we are having the filtration slits, and these slits, they are covered by slit diaphragms, and this space, it is the filtrate in the capsular space. Now the glomerular basement membrane, it is very thick, and that is about 300 nanometers and made up of three laminas. The central, we call it as an electron dense layer, that is lamina densa. Then inner and the outer electron lucent layer, that is the lamina rara interna and the lamina rara externa. The lamina densa, it contains the network of collagen type 4 fibrils and thus acting as a physical barrier. The electron lucent layers, they contain the glycosaminoglycan heparin sulfate, which bears the negative charge. So the glomerular membrane is therefore both a physical barrier and an electrical barrier to the passage of the large molecules. Now this Bowman's capsule, it leads into the renal tubules, and it consists of the proximal convoluted tubule. Then we are having the loop of Henle, and then we are having the distal convoluted tubule. So in between the proximal convoluted tubule, we are having just at its beginning or at the junction of the glomerular capsule, there is narrowing, and we call this area as the neck. And this neck, it is lined by simple squamous epithelium as it is in continuation with the glomerular capsule. The proximal convoluted tubule, it is made up of an initial part having many convolutions. That is, this area, it lies in the cortex. And then we are having the terminal straight part that descends into the medulla to become continuous with the descending limb of loop of Henle. The cytoplasm, it stains pink in H&E staining. The cells or the lumen of the proximal convoluted tubule, it is very less, that is small lumen. It is lined by simple cuboidal epithelium. Then this luminal surface, it is provided with a brush border, and we can see this brush border under the light microscope, and here we can see these cells and their apparatus, that is the luminal surface, it is covered by lot of microvilli. The basal surface of these cells, or the basal surface of the epithelium, it shows the striations which are due to the infoldings of the plasma membrane and the longitudinal placement of the mitochondria in between the two basal foldings. After PCT, it is continuing as the loop of Henle. Now this loop of Henle, it is having the descending limb. So this descending limb has a short thick segment which is straight part of the proximal convoluted tubule, and then we are having a long thin segment. This long thin segment of the descending limb, it continues now as a loop of Henle, and there it forms a ascending limb. This ascending limb has a short thin segment and a long thick segment which is a straight part of the distal convoluted tubule. Now the structure of the segments of the descending limb, we can see here. The descending limb of the PCT, the thick segment of the ascending limb, it shares the structures of the distal convoluted tubule. These thick segments, they are having the cuboidal epithelium, whereas the thin segment of loop of Henle, they are lined by simple squamous epithelium. The ascending limb of loop of Henle continues as the distal convoluted tubule. So the straight part that is continuing with loop of Henle, it is straight, and then we are having the convoluted part. This convoluted part at the terminal end of the DCT again becomes straight, and this part is the junctional tubule or the collecting tubule and ends by joining the collecting duct. And the epithelial lining here, it is the cuboidal epithelium. So PCT and the distal convoluted tubules, they can be distinguished between. So here we can see the PCT, the lumen is less, whereas in the DCT lumen is more. The cuboidal cells lining, they do not have the brush border in the DCTs, and they are less stained. So then let us talk about the collecting tubules. So the collecting ducts, they begin as the continuation of the distal convoluted tubule. As it enters into the medulla, it is joined by several other collecting tubules to form a large duct of Billini or the papillary duct, which opens into the apex of the pyramid, that is the renal papilla. Here we can see on H&E staining the collecting tubules. Here you can see these are the collecting tubules lined by simple cuboidal epithelium with distinct cell boundaries and the cell cytoplasm. The papillary ducts, their cells, they are larger, wider, and they are lined by tall columnar cells with a pale cytoplasm. We are having a group of specialized cells near the glomerulus of each nephron, and we call it as the juxtaglomerular apparatus. It is formed by the macula densa of the distal convoluted tubule. Then we are having juxtaglomerular cells of the afferent arteriole and the extraglomerular cells of Pulcicin, or we also call them as the Lesses cells. Now the macula densa, these are the modified cells of the distal convoluted tubule. The walls of the distal convoluted tubule comes in contact with the afferent arteriole, and the lining cells of these, they are densely packed, and it seems that these cells, they override each other because of the densely packing. So we can see this area as a dark area, and the cells, they become columnar rather than cuboidal, The nuclei are so densely packed that they overlie each other. So we call it as macula densa. These cells lie in close contact with the juxtaglomerular cells. Now, if the blood pressure is too low, what happens? The cells of the macula densa, they release the adenosine, which causes the vasoconstriction of the afferent arteriole. This will slow down the glomerular filtration rate, so the less water is lost and the blood pressure it increases. Now the smooth muscle cells in the tunica media of the afferent arteriole, they are modified to form the juxtaglomerular cells. They are large rounded spherical cells with the nuclei, now situated very close to the macula densa, and they are innervated by the adrenergic nerve fibers. On electron microscopy, these juxtaglomerular cells, they are having a characteristic feature of a protein secreting cells, and the secretory granules within these cells, they secrete the enzyme renin. They are sensitive to blood pressure in the afferent arteriole, that is, we also call them as the mechanoreceptors. Now, other than macula densa and the juxtaglomerular cells, we are having the other group of cells, that is the extraglomerular cells. We also call them as the Lassus cells or cells of Purkinje. They are near the juxtaglomerular cells and they present like a lace-like network. So that is why we call them as Lassus cells. So here we can see the juxtaglomerular apparatus. We are having the macula densa, that is columnar cells of distal convoluted tubule. Then we are having juxtaglomerular cells having the modified smooth muscle cells of the afferent arteriole which secrete renin, and the Lassus cells, they are the modified mesangial cells, and their processes, they form the lace-like network. So what is the mode of action of the juxtaglomerular apparatus? So whenever there is decrease in blood pressure or decrease in sodium concentration in the blood, there is stimulation of juxtaglomerular cells. They release renin, and after this secretion of renin affects the angiotensin in the circulating blood, which converts it into angiotensin 1. So in the presence of angiotensin converting enzyme in the lung, angiotensin 2 is formed. So this leads to the secretion of aldosterone from the adrenal gland. So when aldosterone is secreted, so there is increased reabsorption of sodium and water. So there is increase in blood volume and the blood pressure. So this cells, that is all the PCTs, DCTs, glomeruli, they all are to be present and supported by interstitial space. And this space, that is the renal interstitium in between the tubules and blood vessels, it occupies a very small volume in the cortex, but it is increased in the medulla. So it is formed by the connective tissue with fibroblasts, interstitial dendritic cells, collagen fibers, and ground substance which is rich in proteoglycans. Whereas in medulla, the secreting interstitial cells are present and they help in the synthesis of prostaglandins and prostacyclins. So on H&E staining, here you can see this is the outer cortex and the inner medulla. So in the outer cortex, we can see the glomerulus along with the Bowman's capsule and the capillaries. So we can see here the two layers, that is the outer parietal layer and the inner visceral layer, that is present in close adherence with the capillaries. So these are proximal convoluted tubule with simple cuboidal epithelium with the brush border. Here you can see the difference between the PCT and the DCT. DCT, it is lined by simple cuboidal epithelium. Then we can see here the thin segment of loop of Henle. Then we are having, this is the proximal straight tubule, that is thick descending limb. This is thick ascending limb, and you can see the collecting duct with a large lumen, and here you can see in between we are having the capillaries. So here you can see the difference between the distal convoluted tubule and the proximal tubules, the lumen, the size of the cells and the shape of the cells, and the brush border. Here you can see these are the large collecting tubules, along with this is small loop of Henle, and this is one glomerulus. This is a section of the kidney stained with hematoxylin and eosin stain. So here we can see this is the outer cortex and this is the inner medullary area. So this is the glomerulus along with the glomerular capillaries, and here you can see the Bowman's capsule. And then we are having different PCTs on the cut section. This is the proximal convoluted tubules, the smaller in size and darkly stained tubules. Then we are having the this large one, you can see these are the descending segment of the proximal convoluted tubule along with different vessels. We can see we are having the glomerular arteriole over here, and here you can see this is the juxtaglomerular junction that we can see here the PCT along with this glomerulus. So then we are having the straight ascending segments of the distal tubules. These large one, they are the collecting tubules, and these thin segments of the loop of Henle can be seen over here. This is a slide of the kidney cortex showing the juxtaglomerular apparatus. Here we can see this is the distal convoluted tubule, and these are the specialized cells, that is the macula densa of the distal convoluted tubules. Here we can see the juxtaglomerular cells of the afferent glomerular arteriole, and this is the glomerulus along with glomerular capillaries, and this is the parietal layer and the visceral layer of the Bowman's capsule. Along with this juxtaglomerular apparatus, we can see the other cells also. Here we can see the proximal convoluted tubule with centrally placed darkly stained nucleus along with the brush border of these cells, and here you can see the distal convoluted tubule with the large lumen, and the cells again they are cuboidal in shape with centrally placed nucleus. And here we can see the interlobular blood vessels, that is the venules and the arterioles. So here you can see this is a large collecting tubule along with its basement membrane.